I'm not exactly sure how I arrived here as your keynote speaker today, and we were speaking earlier this morning. It's a very unique experience for me as a supervisor and a leader within the FDA to get to speak to such a large group of patients. So for me, this is really a privilege and an honor today, so thank you so much for having me here today. As was just mentioned, I started this journey into pulmonary fibrosis almost 20 years ago as a pulmonary fellow, where I was doing basic lab research in a lab injecting mice with bleomycin. And no offense to the mice, but I much prefer this forum today. So thank you so much again for having me. So I must be where I was meant to be, because 20 years later, here I am in this room with you. So today, as was mentioned, the title of my talk is Regulatory Considerations for Developing New Treatments While Maintaining Clinical Trial Safety. And I know a lot of what you hear about with FDA is the new FDA-approved medication. But there's so much more to what we do, and safety is such a huge focus of what we do. So I hope that I can give you a flavor of that during my talk today. And I thought it might be useful before going into specifics to sort of provide a general overview of what we do, because even after 20 years at the FDA, my mother still asks me questions about what I do, and will say things like, I have this pain in my ear. Can you go research it and come back and tell me how to fix it? And so I think that's our fault as regulators. I think we're not transparent about what it is we do at FDA. So I'd like to give you an idea of that, and then I'll sort of transition into general clinical trial safety and also specifics of pulmonary fibrosis programs. So with that, we can get started. I have no disclosures because I work for the FDA. So here we go. This is the outline of my presentation this morning. So I want to provide a general overview of FDA's role in drug development. We're constantly interacting with various stakeholders, industry, patients, investigators, and we're providing a lot of input and advice into the review of efficacy and safety of drug products. I want to then provide an overview of how FDA maintains the safety of patients in clinical trials for pulmonary fibrosis, and then close with a few future directions and some concluding remarks. So this is a very large organization, as I'm sure many of you know. We're 7 centers at the FDA, so it's always funny to me when someone says, oh, I know someone who works at the FDA. I probably don't know them, but I work in the Center for Drug Evaluation and Research, and within the Center for Drug Evaluation and Research, I work in the Office of New Drugs, and we're very much set up like a clinical practice or a hospital system would be set up. We have various divisions that are in charge of the different disease areas in medicine. So I'm specifically involved in pulmonology, allergy, and critical care. So everything I'm going to talk about today in terms of patient safety and clinical trial safety really stems from the FDA mission. And I won't read this whole slide, but I want to emphasize that we really are focused on ensuring the safety, efficacy, and security of human and veterinary drugs, biological products, and medical devices. So this is our main focus, although as we sort of move on later in drug development, as I'll talk about, efficacy comes into play as well. So as I mentioned, and I've told my mother several times, we don't develop the drugs ourselves. We interact with sponsors and investigators throughout the life cycle of the drug, which is displayed here on this slide. And really, the specifics of phase 1, phase 2, phase 3 are not that important here. But really, what I want to emphasize is that sort of early on, I guess, do I have a? Oh no, I guess maybe not. Okay. Early on in development, sort of towards the left-hand side of this slide, research development, preclinical development, and early phase development, the focus of drug development is really on safety. Yes, we want to have some information that potentially this drug is going to later work, but really what we're worried about and focusing on is safety. First in animal models, and then going forward into human subjects and patients. And so this, while the focus on safety really remains throughout clinical development, it's really a focus during the forefront of development. And then we come back to it at the end when we have marketing applications. And I'll talk a little bit more about that. So initial sort of investigational new drug applications, which are on the left-hand side of this slide, we're really focused on safety, and then efficacy comes into play as we get into the marketing applications, the new drug applications, or the biologics licensing applications. So how do we provide this advice? If you ask my son what I do, he will tell you that my mom has meetings all day. That is true. We are constantly meeting with stakeholders to provide advice. And this is highly regulated, and also there is a guidance. So one of the ways that FDA provides advice is through these guidance documents. So throughout this talk, you'll see various screenshots of guidance for industry. And so sponsors and developers can request meetings with the FDA throughout drug development. And we provide feedback in response to specific questions. We engage in teleconferences, video conferences, and face-to-face meetings. And there are different meeting types and different timelines that we have to adhere to respond to our stakeholders. And these depend on the differing stages of development and other factors, which I'll go into in a little bit. But everything is documented throughout this. So there are no sidebar conversations and agreements. Everything is very strictly documented, and these things are captured in responses and meeting minutes. So specifically to the pulmonary fibrosis indications, such as idiopathic pulmonary fibrosis and progressive pulmonary fibrosis, for which we see a lot of applications. These are obviously serious and life-threatening conditions for which there exists an unmet medical need. And these types of indications can qualify for what we call expedited programs for serious conditions. And again, I've included a screenshot of a guidance here, but there are various expedited programs. There's fast track designation, there's breakthrough therapy designation, and then finally there's priority review. And what these really allow for, there are different criteria to fulfill each of these, but what these really allow for are frequent interactions, more frequent interactions with FDA, faster response times in some cases, and finally, if a product is to be granted breakthrough therapy designation, it allows for a faster review once it's submitted for a marketing application. Now, I will say that we exercise some discretion here. If we see the unmet medical need and a product doesn't have breakthrough therapy designation, that doesn't mean, we say, oh, we're going to drag our feet on this one. So, but these are mechanisms that sponsors have in order to increase the frequency of their interactions with us at FDA. But we don't only meet with industry. We also try to engage our patients. And this is a part of the Patient-Focused Drug Development Program, or PFDD. And this was a commitment under the PDUFA 7, which is the Prescription Drug User Fee Act. And the Patient-Focused Drug Development Program offers a more systematic way of gathering patients' perspectives on their condition and treatment options. And we feel that these meetings can help advance a systematic approach to gathering input. These are important for us to establish the therapeutic context, and because this is an important aspect of the benefit risk assessment, which I will go into in subsequent slides. And we understand that you as patients are uniquely positioned to inform understanding of this context. So I don't know if I saw any of you back in 2014. We had a meeting, it's been a while now, in September of 2014, and we were given the unique opportunity through this program to hear directly from patients, patient caregivers, and patient representatives about their experiences with IPF and its treatments. And the discussion focused on two key topics: the symptoms that matter most to patients and their impacts on daily life, and the patient's perspectives on current approaches to treating, and this was specifically for idiopathic pulmonary fibrosis. For each topic, a panel of patient and patient representatives shared comments to begin the dialogue, and we invited comments from the audience. We had folks joining over webcast, and we also asked a number of polling questions. What we really were looking for was what mattered to patients. And this is what we heard, and I don't think this will be a surprise to anyone in this room: coughing, shortness of breath, fatigue, impacts on daily life, activities, and burdens of oxygen therapy. We published what we heard and saw in a document called The Voice of the Patient, and the link is provided in this slide. And we've had a series of these patient-focused drug development programs for different diseases that all get published in this way. And we are very careful to use exactly what the patients tell us in terms of what they're reporting in terms of their disease. So I mentioned that this was back in 2014, and so this slide, what have we learned, but really what are we continuing to learn? And I'm going to highlight this a little bit later in the talk. Patients' chief complaints may not be factored explicitly into drug development programs, including the measures of drug benefit planned in trials. And patients want to be as active as possible in the work to develop and evaluate new treatments. And it was very eye-opening to me that really, patients such as yourselves weren't really expecting FDA to address all the gaps in current treatment or approaches to treatment to drug development, but they did want FDA to help identify the most effective pathways for them to play a major contributing role. And so I presented this at the American Thoracic Society now probably 6 or 7 years ago, and I know that what we have done with this information is not enough. We need to continue to do more, and I think one of the things that I'll highlight later is why this might be coming to the forefront even more now. So with that, I'm going to transition into patient safety because right from the patient experience to patient safety is a nice transition. As I said, this is one of our primary focuses at FDA when we review submissions for new clinical trials and new drugs to treat pulmonary fibrotic diseases. So I'm going to give you a general idea of some of the things that we do, and this sort of spans all clinical development programs, and then I'll come back to some specific things that we do in pulmonary fibrosis trials to ensure the safety of our patients. So if you think back to that diagram that I showed of the drug life cycle of sort of the preclinical work, these are 2 slides that I'm going to go through, not in a lot of detail because they're very highly technical in terms of what's required of animal studies prior to even going into humans, but I felt that this was important because this is one of the ways that we ensure the safety of patients in early clinical trials. So we don't just go right into patients with drugs. We start off with a series of animal studies, the most important of which on these slides are really the general toxicology studies, which are the sort of the 4th one down. And these general toxicology studies provide us with what we call safety margins, a safe dose to proceed with in humans. The proof of concept and the other safety studies are important, but these are the ones we really rely on early in our decision making to decide what dose is safe to give to our patients early on in clinical trials. The other reason that these programs are important before we go into patients, they help us identify what we need to be looking for in terms of our safety monitoring. If we see a liver toxicity or a GI toxicity in the animal models, we know that we have to pay very specific attention and potentially incorporate more thorough safety monitoring around those aspects when we go into clinical trials. And this is a distinction that is a little technical, but whether the molecule is a large molecule or a small molecule, we have different requirements for the studies. But it's needless to say, regardless of sort of the specifics here, we really exercise flexibility because we understand that the pulmonary fibrotic diseases carry a large unmet medical need. And so we really look at the benefit risk to calculate how much of this is necessary at different times in development. For example, the carcinogenicity studies, which are looking at whether the drug can actually cause cancer, are usually required before a marketing application or before late phase studies in a lot of different drug development programs, but for IPF and PPF, we exercise some flexibility as to when these are required so that we can sort of accelerate development. The other thing that we pay a lot of attention to is lung toxicity, because as you can imagine, it doesn't make much sense to treat a lung condition with something that could potentially also cause a lung toxicity. So we're very mindful of lung toxicity in these programs. So moving sort of to the right on that diagram, when we get to our sort of our first submission, our investigational new drug or IND application, one of the things that we at FDA pay very close attention to is the investigator brochure and the informed consent. Patients need to be informed and investigators need to be informed to make an informed decision about their own safety in clinical trials. And the investigator's brochure will contain a summary of the pharmacological and toxicological information of those animal studies that I mentioned in the previous slides, and to the extent that it's known, the effect in humans. And it should also contain a description of the possible risks and side effects in previous clinical trials. So when we're evaluating these new submissions, we have the ability to not allow them to proceed if the safety is not there. And one of those criteria is a misleading investigator's brochure. So if that information is misleading in any way that could compromise the safety of patients, we ask that that be amended before the clinical trials are allowed to proceed. Again, at this very early stage, in this investigational new drug stage, safety reporting is paramount. And so before I go into what we require for safety reporting, I thought it would be important to go over some of sort of the jargon that we use. So on the next two slides, very quickly, I'll just go through adverse events, suspected adverse reactions, and adverse reactions. And so here we see an untoward medical occurrence associated with the use of a drug, something that we think is due to the drug, and then any adverse event that's caused by the drug. And also, we pay very close attention to things that are unexpected, not listed in the investigator's brochure, things that are serious. A serious adverse event is actually defined in our Code of Federal Regulations as being death, a life-threatening adverse event, an inpatient hospitalization, persistent or significant incapacity, or a congenital anomaly or birth defect. And again, we pay very close attention to these life-threatening events. And there are statutory requirements for sponsors to notify FDA in a written safety report of any of these adverse reactions. And there are different timelines that depend on to the severity of these reactions. We also encourage the use of data monitoring committees, which is a group of individuals with pertinent expertise that reviews on a regular basis the accumulating data from one or more clinical trials. And the DMC advises the sponsor regarding the continuing safety of the trials and the trial subjects and those yet to be recruited. Now, these DMCs are more important as we sort of get into later phase development, or if there's a large safety concern that we've noted early on in the development of the product. So this is a sort of maybe not the most intuitive thing for safety, but dose ranging is something that is very important for safety, in my opinion. It usually gets tied to whether which dose is effective, but exploration of more than one dose is encouraged in all of our clinical programs. And this is important because it allows for collection of not only efficacy information, but controlled safety information. And it can inform safety monitoring and adverse events of special interest for future trials. So as we sort of go through and go into later phase development, we can decrease the number of treatment arms in the pivotal trials. And really, this goes to patient safety because it reduces the chance that you as patients are exposed to an ineffective or an unsafe dose over a longer period of time. And we like to see the dose ranging earlier to avoid the failure of pivotal trials due to choosing the wrong dose. We never want to end up in a setting where we investigate a dose that we think maybe at a lower dose wouldn't have had, the safety profile we see, but we don't have information on a lower dose. So this is very important, not only to efficacy, but also very much to safety. Standard of care has been an evolving sort of conversation since the approval of the pirfenidone and nintedanib programs back in 2014. So once we had approved therapies for idiopathic pulmonary fibrosis and nintedanib for progressive pulmonary fibrosis, there were ethical issues of withholding available approved therapies for longer-term trials, and this posed a safety concern to our patients, especially if trials went on for longer than 6 months duration, which many of these trials do. So initially, we didn't really know how these drugs were going to be used. So at FDA, we were very prescriptive. Everyone must be on them. You can't be taken off of them. And we really we prohibited patients from entering a trial without them. But we sort of gained an understanding that not everyone was going to be on these medications. We saw over time how they were used. And so we sort of pivoted to leave the standard of care to the investigator, or the provider, or the patient discretion. And we make sure in our protocols that we review that patients should be able to start on these therapies at any time, so that they shouldn't be restricted just because they're in an investigational trial. In terms of drug-drug interactions, a lot of our patients are on these two medications already, and we really encourage early investigation of whether these two medications will interact with the investigational treatment, because these could create safety concerns. If the exposure to the investigational treatment goes up, it's really a higher dose than maybe what the safety margins were allowing for. So again, this really informs the use of background therapy and can inform the dose that may be used going forward for the investigational product. We require long-term safety information, and it's 52 weeks. This is recommended in our guidelines to have 100 patients, at least 100 patients for 1 year in terms of having this 52-week safety information. We can borrow from other indications if the drug is being used, if that's relevant, but we really like to see at least 100 patients for a year. And again, this is our opportunity to obtain controlled safety data where we're collecting all the information, we're comparing to a control group, because this is really important. It's understanding the safety profile over time so we can inform our healthcare providers and patients to make informed decisions about their treatment. It's also really important because if we look at sort of important clinical outcomes that may not be apparent early on, we find some disturbing things sometimes. And so this is a recent study, the Isabella study, that was stopped because survival actually went in the wrong direction. And so we really have to be mindful that sometimes we're measuring something and we're actually might not be able to measure something else if we don't go out long enough in our clinical trials. So we can't really talk about safety without talking about efficacy. They're very closely linked because all drugs can have adverse effects. So we can't look at safety in a vacuum. We have to balance it with the efficacy of the drug. And that goes into what we call the benefit risk assessment. And this is something that we're doing over the life cycle of the drug, but it really becomes very prescriptive when we get to approval. And it's required in our reviews for every marketing application. So just a quick word on forced vital capacity. This is the endpoint for efficacy that we've used in the 2 approvals of pirfenidone and nintedanib. It's established somewhat of a regulatory precedent, and most programs are now using this as well as their efficacy endpoint. There's a biological plausibility, there's an association with mortality, but there are challenges and limitations. And especially with the approved therapies, and folks already being on them, it's harder to show a benefit on top of standard of care. So this may not be a feasible endpoint for very much longer, and it's also potentially not a measure of how a patient feels or functions, which is very, very important. So this guidance document was just newly published. It's the benefit risk assessment for new drug and biological products. And it really brings patient experience into the benefit risk assessment. I think this is so important, and I think this is where we need to be going with this field, but we still aren't quite there yet. We need to gather stronger evidence to support use of more patient-centered endpoints, such as patient-reported outcomes and functional assessments, so that we can really understand the therapeutic context of the disease and how it affects our patients and how they feel and function. So I think I went a little bit over, but we're almost finished. So thank you so much. I just wanted to summarize by saying we understand at FDA the unmet medical need in IPF and PPF and other pulmonary fibrotic diseases. We acknowledge the need for better endpoints, and we're working hard to get there. We know we need new treatments. The treatments we have are not enough, and we can't emphasize enough, I hope that I've emphasized, that maintaining the safety of our patients in clinical trials is really of the utmost importance at FDA. And we really do feel that we want to encourage the development of endpoints based upon how a patient feels or functions, understanding that this has to be done with the proper rigor to answer the questions and do the greatest service to our patients. So in closing, I think one of the most important ways we can protect the safety of patients is not compromising on the efficacy standard required for approval. As regulators, we don't forget about the human aspect of the disease. We are trying to understand, regulate, and make decisions regarding drug approval. And as patients and clinicians, we also ask that you remember that there's a human aspect to this decision making. I hope that I've provided a human face to that decision making today. And at the end of the day, we're scientists, we're physicians, we're trying to interpret the data in a regulatory context within the confines of our regulatory processes and really do the best that we can for you as patients. And there's an element of judgment and an element of reasoning involved, and we don't make the decisions in a vacuum. We engage experts, we enlist help from outside sources so that we can understand that the data can be interpreted in a number of ways. And we respect all of these interpretations, even if we don't agree with them all the time. So we make the best decisions that we can with the data that we're given. And so we need our patients to help us. You're our biggest collaborators. Keep enrolling in clinical trials, because this is one of the best ways that you can advance the development of drugs for pulmonary fibrotic conditions, and we at FDA will do our very best to keep you safe in those trials. Thank you.
My pleasure to be here. Myself and the National Heart, Lung, and Blood Institute has had a long-standing partnership with the PFF, and it's my absolute pleasure to continue that tradition today. So my job is to tell you about the research that the NIH and really the NHLBI has supported in recent years, give you some highlights across the spectrum of research that we support, and also give an eye towards the future and where we think the research is going to go in this field. See if this will advance. I also don't have any conflicts because I work for the NIH, and as some of us were joking earlier, if you don't believe me, you can actually FOIA my government financial report so you can confirm that for yourselves. So let me cut right to the chase. So usually when I'm in the room, the first question I get is about money and how much of it the NIH is spending on disease of interest, in this case pulmonary fibrosis. And I'm happy to report that the answer is a lot. And in 2022, it was $131 million that the NIH put towards pulmonary fibrosis research. It turns out that you now can verify this and track this yourselves. So as of 2022, I was part of an effort that got pulmonary fibrosis added to a list of what we call research condition and disease category, or RCDC terms. And these are publicly reportable terms, research areas that we inform Congress annually now on the projects that we have funded and the overall spending in this space. And so now, as you can see here, between psoriasis and pulmonary hypertension, which was another new area that we added this year, pulmonary fibrosis is now on the list. And so I think this is really important. It's also important because Dr. Rahage's predecessor, President and CEO of the PFF, Bill Schmidt, used to annually ask Congress for this number, and then Congress would ask me for this number, and I'd have to manually generate it. And so I'm quite happy now that a computer algorithm does it for me. So that's sort of the high-level top-line number is 131 million in 2022, but it turns out we have an ancillary database called NIH Reporter that will give you more detailed information about the projects that the NIH funds. And it'll break it down for you by the individual institutes and centers, or ICs, such as NHLBI, and there's some information about each of those projects that you can get. So there's a summary, an aims page, per se, for each of these projects. You can see the institution that received it, how much money, the principal investigators. And there's a number of different visualization tools in NIH Reporter. So as you can see, you can get this in table format, you can get bar graphs, you can get pie charts, and you can see both the total number of projects and the total amount of spending. And I'm quite proud to say that the number one institute, as you might imagine, is my institute, which is NHLBI. In 2022, we funded 182 projects that totaled about $89 million. The other tool that you can see in there is this graphic to your right, which is sort of a summary of the key terms that come out of these projects. And so you can see that we are funding a number of different clinical trials. There's been a focus on animal models. There's some basic science focus on things like epithelial cells that are important for the development of fibrosis. And so you can use that visualization tool to get an idea of the types of projects we're funding in terms of disease areas. So NHLBI's overarching goal is really to support a robust pulmonary fibrosis portfolio across the research spectrum. And what I mean is from basic science to phase 3 multi-center clinical trials of novel interventions. And the way that we do that at baseline is that we have a series of what we call investigator-initiated funding mechanisms, things like the R01 and the P01 that really support basic science research. We now have a couple of different programs that support translational research, including what we call the Catalyze program that I'll tell you more about in a minute, and our small business program. And we also have a series of funding opportunities for clinical trials. And again, I'll tell you a few more details about those opportunities. And then, of course, underlying all of these areas is our interest in maintaining a strong workforce that does conduct pulmonary fibrosis research, and we do that through support of graduate and postdoctoral trainees through our F&T mechanisms, as well as early career investigators who are looking to establish themselves as independent researchers, which we do through our career development or K awards. So just to give you a few highlights within each of these areas across the spectrum, I want to highlight a program that I helped to develop, which is this Advancing Novel Research Models to Study IPF, which supports 5 different cooperative agreements. One to Daryl Cotton's group at BU that's focused on using inducible pluripotent stem cells to help develop organoids and model pulmonary fibrosis. Mike Beers' group at University of Pennsylvania has a series of transgenic mice that carry surfactant protein C mutations that spontaneously develop fibrosis. Bridget Gompert's group at UCLA has another in vitro model organoid system. The 4th project is from Steve Rowe's group at UAB, which is developing the use of ferrets to study both pulmonary fibrosis at baseline, but also the role of MUC5B in the development of fibrosis. And then lastly, Dan Schumperlin's group at the Mayo is using precision cut lung slices and other cellular systems to model IPF. And our goal here was really to support a range of complementary models that themselves each recapitulate at least one important disease-defining feature of IPF, recognizing that the most commonly used model, the bleomycin model, is maybe not the best model to really study pulmonary fibrosis. And so our hope is that collectively these models together represent sort of one larger model system that investigators can use to sort of tap into the specific research question they have. And I've just highlighted here that there's been a collaborative project that's come out of this between 2 of the groups, so Dr. Cotton's group and Dr. Beers' group, showing that you can use these patient-derived inducible pluripotent stem cells to study in human cells the information that Dr. Beers' group had learned in mice. And so really what we're seeing is a merging of these models into what I think is a really powerful tool for understanding disease pathogenesis. Transitioning to thinking about how we support translational research, I mentioned that we have what's called the Catalyze program. The goal of this program is to support both academic researchers and small businesses up until the point that they're ready to submit an application to Dr. Karim Shah's office at the FDA for a drug approval. And so what we support is both product definition, which is really early phase translational work to identify disease targets, identify lead series or lead compounds or devices for testing. And we now have what's called a preclinical platform that takes that a step further and helps to develop those drugs and devices and do some of the necessary pharmacology, toxicology, chemistry, manufacturing work that's necessary to apply for regulatory approvals. We have a number of different funding opportunities within the Catalyze program. One is to really enable technologies and platforms to identify targets, and so this is really sort of a screening tools for identifying novel drug candidates. We also have a paired couple of funding opportunities that are focused on small molecules and biologics, and then we have 2 other ones that are focused on devices. And so collectively, we have funded a number of projects out of these funding opportunities that are focused on pulmonary fibrosis that I'll tell you about in a minute. And I just want to highlight that applicants who are funded through this program get access to a coordinating center that's run out of the Research Triangle Institute that helps lend business development and regulatory support for these applicants to help eventually get their drugs and devices to market. So we funded 3 projects within the Catalyze program that are focused on pulmonary fibrosis. One is a PET probe that targets oxidized collagen to help identify early fibrosis in the lung. This has been developed by Peter Caravan's group at the MGH. This project is actually finished and it's now, I think Peter's getting ready to try to take this into human studies. Another project that was recently funded is focused on the development and use of a fibrosis on a chip system that uses basically a microtissue system that we think can be used to help identify new drug candidates in the pathways that they target. And then lastly, another recently funded project is focused on the development of YAP-TAZ inhibitors that we think may be promising therapeutics for the treatment of fibrosis in the future. And then lastly, across this spectrum of research, we have a whole suite of clinical trial funding opportunities. And these range from early phase, phase 1 safety trials, to pilot studies that get people ready to do phase 2 efficacy studies, as well as those phase 2 and 3 efficacy studies themselves, both single-site and multi-site trials. And then lastly, we also have what we call mechanistic clinical trials, which are really trials to understand the mechanism of action for interventions that seem to have some preliminary support of safety and efficacy data. So really, whichever flavor of clinical trial you're interested in conducting, NHLBI has a funding opportunity to support that work. Under these funding opportunities, we have supported a number of clinical trials which are still ongoing. One of which many of you are familiar with because it represents a partnership with NHLBI and the Pulmonary Fibrosis Foundation, as well as the Three Lakes Foundation, is what we call the Precisions Trial. The Precisions Trial is, I'm sure you'll hear more about it at this meeting, is a novel pharmacogenomic precision medicine trial that is enrolling IPF patients who carry a specific gene allele that's derived from the TALB gene. And prior studies had suggested that those who carry this allele may be more likely to benefit with treatment with the antioxidant N-acetylcysteine. And so the trial is enrolling specifically those patients and comparing it to placebo. And I'm sure Imre and Fernando would like me to tell you that they are right on schedule with enrollment. It's set to finish at the end of this year. And the other thing I want to highlight about this study is that it's not just a clinical trial. It's actually much more than that. It's also leveraging samples obtained from the PFF's patient registry to do molecular analyses to really understand more about what are the genetic risk factors for this disease, how can we use that information and those biomarkers to improve diagnosis, as well as the prediction and understanding of the mechanisms that drive progression. And so this is a really important program for a number of reasons. One, it's a clinical trial, but two, it highlights really the value of the data in the samples that the PFF registry participants put into that, because it gets used for far more than just sitting in a database. It really helps to drive the research. We also have a number of other trials that we're funding. We have a phase 1 trial of a compound that we fortunately call EGCG rather than its full name, which is a multi-center placebo-controlled phase 1 safety trial. So this is in early stages, but there's at least some preliminary pilot data that was published in the New England Journal that suggests this may be an efficacious treatment for IPF. And then we have 2 trials from Steve Duncan at UAB, one that's focused on a novel compound that inhibits NOX4. So this is targeting oxidative stress pathways in IPF. As well as a second study that's focused on trying to reduce the severity of exacerbations for those who are living with IPF. And so this is a clinical efficacy trial. So in thinking about the future, we've obviously made great strides. We know a lot more about IPF than we did a decade ago when we last held a workshop to try to identify key areas for research that the NIH wanted to advance on this topic. We went back about a year ago and revisited this, and what came out of this workshop was the recognition that we need a few things moving forward. We need better models and research tools to study pulmonary fibrosis and uncover new therapies. We need better early disease and progression risk factors and biomarkers that we can use to improve diagnosis and monitoring of patients. And we need to be innovative in our thinking about clinical trial design, recognizing that this is a relatively rare disease and we really want to maximize the output of our trials for our patients. And so I won't get into a lot of the details here as I'm running out of time, but just know that a workshop report is coming soon. And you'll all be able to see all the details about what was discussed at the workshop and what we think is coming next for pulmonary fibrosis research. I'll also just briefly highlight a couple of different things that we support. Obviously, the goal is to prevent and/or cure pulmonary fibrosis, but we're not there yet. And until we are, we recognize there are a few other needs in this community, notably oxygen delivery, which many people tell us that their needs are not met for a number of different issues. And so one of the things that we had done in partnership with the NSF, the FDA, and the CMS is we issued this innovation challenge focused on oxygen delivery. And there were 2 phases of this. We were able to award 4 prizes to a number of different companies who are developing oxygen delivery devices. So some that are focused on sort of making the delivery of oxygen more precise, some that were focused on concentrating the oxygen, and some that were focused on sort of the portability of oxygen. So we were happy in February of last year to award 3 winners and an honorable mention for these devices. And now they have prototypes, which we hope that they will continue to develop and eventually take to market. The other thing that we recognize is that there is an existing cure for pulmonary fibrosis, and that's lung transplantation. However, lung transplantation outcomes could certainly stand to be improved. Right now, the average survival after lung transplantation is about 6 years. The vast majority of patients who get a lung transplant now are those with restrictive lung disease, and the vast majority of those are patients who have pulmonary fibrosis. Nowadays, it's almost 3/4 of lung transplant in the US go to those with restrictive lung disease. However, we'd like to see the outcomes improve for these patients, have them live longer than the average 6 years. So NHLBI recognizes this need, and we are now supporting what we call the NHLBI Lung Transplant Consortium that is focused on a number of different projects around sort of clinical management and selection criteria for transplant candidates to really understand what promotes the best outcomes in these patients. And so this is a national program. It's got 19 participating centers, which you can see on the map here, and we're pretty excited about this program. This is a nation program. It's only been up and running for about a year, so I don't have results to tell you about today, but hopefully at a future meeting I will. So I want to end by just mentioning that there's the lung division that I work in is going through a reorganization. And we are doing this with the hopes of better serving the research community. It's gotten to the point where, as you can see from the list here, that we've had 2 branches in the sleep center in our division. The branch that I oversee has really grown, and it's become somewhat unwieldy. And so our feeling was that we could probably reorganize to better serve the community. And so what's coming in early 2024 is that we are splitting from 2 branches in the sleep center to 4 branches in the sleep center. And what I'm happy to tell you is that one of those branches will be called the Restrictive and Vascular Lung Diseases branch. And so basically 50% of that branch will be focused in part on interstitial lung disease, including pulmonary fibrosis. We are also in the process of revisiting our strategic vision as part of this reorganization process. And a request for information will be coming out in the coming months that'll allow the public, including everyone in this room, to have input on that strategic vision. As part of this reorg, I will actually move to the acute and infectious lung diseases branch, but the continuity that you will have is in our current program officers. So Dr. Christian Gomez, who's here today, who oversees the IPF portfolio, and Dr. Luis Vuga, who oversees the non-IPF ILD portfolio. So those will remain your primary contacts for fibrosis research. And I'm well over time, so I'll end there and say thank you.
Oh, it's really nice to see all of you. It's, as a physician who takes care of patients with pulmonary fibrosis, this warms my heart tremendously. So thank you for being here. In contrast to Dr. Karimi Shah and to Dr. Craig, I do not work for the Department of Defense. I work for the University of Pittsburgh, and I have an opportunity to work in a village. And that's the name of this session. I don't have relevant conflicts. I do report that I have funding from the NIH. I have funding from the Department of Defense, which I think is why I'm here. And I also have funding from Boehringer Ingelheim. So again, it does take a village. And that's something that resonates with me. And I'm going to take an opportunity to really talk about the place where I work. I work at the Simmons Center, which is in Pittsburgh. Anybody here from Western Pennsylvania? All right, go Steelers. So this is, we were founded in 2001 by a generous gift from a man named Richard Simmons, not the Richard Simmons you think of, This was in memory of his wife who died of pulmonary fibrosis. Now, with all the relevant resources in the world, because he's wealthy, his wife did suffer with this disease. And this moved him in a way that he could help other people. He helped establish this center. And this center became a place of patient care. And it was run by our founding director, Dr. Naftali Kaminsky, and Dr. Kevin Gibson, who's our medical director, who you may know is the ATS Clinician of the Year. And many of you here know Kathy Lindell, who's held a number of positions here with the Pulmonary Fibrosis Foundation. And Mr. Simmons charged us with a threefold mission. And the mission was to care for the patients, to support the caregivers, and to perform cutting-edge research. And the only way that this is possible is with a village. And this village here requires everybody. It requires students, it requires PhD researchers, research coordinators, administrators, physician assistants, doctors, nurses, and all the people who make this work. And it's very important because we're all trying to do it for people like you. And so this is an opportunity to work in a village. And so when I think about the title of this talk, to talk about the Department of Defense, I think perhaps a more witty title would be All Hands on Deck. And so this is what the Department of Defense does. And this is it's operated out of the Congressionally Directed Medical Research Program. As you can tell from the logo is the Capitol. The logo is not a military symbol, it's not a weapon. The research comes out of Congress. So the CDMRP is an office that's housed in the U.S. Army Medical Research and Development Command. And it was established by Congress in 1992 as a result of a grassroots effort to fund research in breast cancer. The initial outlay in fiscal year 1993 was $210 million to help breast cancer research. And what's very important here, again, is you can see the importance of grassroots efforts. The research that comes out of the Department of Defense is directed by Congress. And so the CDMRP manages the administration of the grants and the peer review program. And the topic areas, again, are decided by Congress. It's not decided by the DoD, but the DoD's mission often aligns with the subject matters that Congress allocates for this research. And so the CDMRP has a mission. It strives to transform healthcare for the U.S. military, veterans, their families, and the American public. So in fact, it's really striving to provide innovation in healthcare for everybody. And the CDMRP aims to fill research gaps by funding high-impact, high-risk, and high-gain projects that other agencies may not venture to fund. And so that's very important. This is an opportunity to try to do those moonshot projects that may be risky, but may have a high reward in the end. And so to quote the motto of the lottery, you got to be in it to win it. And so it is a funding opportunity that does come out every year. And there's something also very particular about DoD funding, and it's that DoD funding is reviewed not just by scientists, but there are consumer representatives at the review. And so they're people like you. They're patients, they're patient advocates. And it's very important that these research projects not be pie-in-the-sky things, but that they have maybe not immediately, but some benefit to patients in the near future. So one of the line items in the CDMRP docket is the peer-reviewed medical research program. And so this is the largest group. And you can see that this is just a selected number of topics that the DoD funds. It includes arthritis and celiac disease, but then other things which you think might be more relevant for military research, including hemorrhage, malaria, pressure ulcers. But this is where research in pulmonary fibrosis, in overall respiratory health, rheumatoid arthritis, and scleroderma fall. And This is in contrast to the numbers that Matt just put up. This is the total funding of pulmonary fibrosis research over the years of the program. So it's approaching $50 million. So again, it's a drop in the bucket when we consider NIH funding. But again, this is $50 million of research for pulmonary fibrosis that would not otherwise have been spent here. So it does represent a significant outlay from a different federal agency towards helping patients with pulmonary fibrosis. There's over $15 million for scleroderma research and nearly $30 million for rheumatoid arthritis research. And so the CDMRP has funded a lot of very important studies in the history of the pulmonary fibrosis program. And my apologies if your article is not up here, but I want to take an opportunity just to highlight some of these studies. Again, these are studies that have been published in high-impact journals and have made large impact in the field, including this one, which was the first of the single-cell RNA-seq studies to emerge in pulmonary fibrosis. And we see other studies here. This study highlighted the tremendous importance of the macrophage in the pathogenesis of pulmonary fibrosis. This studies MUC5B, the so-called IPF gene, and its importance in the pathogenesis. And so a number of very important studies have come out of CDMRP-funded research. And what I want to sort of conclude here on this slide, again with so many different studies here, is that some of the research actually does lead to work that's important for our military. And this document is one of our ATS documents. It's a guideline. It urges more funding, but this is for the respiratory health of service members who serve in Southwest Asia and Afghanistan. There's a particular group of illnesses that relate to this group, and CDMRP research has gone to fund efforts to try to codify the important issues there. So I'm going to take a couple of minutes here to talk about a little bit of our research, which is focused by the Department of Defense. I'm proud to say that this study is hot off the presses. It came out this past week in the European Respiratory Journal. We've been interested in a molecule called CXCL6. We discovered that in the bronchoalveolar lavage. So the bronchoalveolar lavage is fluid that you can get from the lung that's obtained at bronchoscopy, and that this fluid can be important for detecting potential biomarkers of the disease. And so what we discovered is that CXCL6 at elevated levels in the bronchoalveolar lavage is associated with increased mortality in patients with pulmonary fibrosis. You can see here on the right, these are called Kaplan-Meier curves. Kaplan-Meier curves look at survival of patients. Each curve, these were two cohorts that were done at Hanover Medical School in Hanover, Germany. And there's a cohort done in 2018 and a cohort studied in 2021. The red line shows reduced survival in patients with high levels of CXCL6 in the bronchoalveolar lavage. And so we think this is an important biomarker, but it's not just a biomarker. We think it's actually very important for this disease. On the left here is a Western blot. A Western blot is a stimulation of cells, and you look at the cells in the end to look at certain levels of different proteins. And what we took here were fibroblasts. Fibroblasts are the cells that create matrix and create fibrosis. One of the principal components of fibrosis is collagen. When you stimulate IPF fibroblasts with CXCL6, what happens is at the top here, you can see that there are increased levels of collagen. And when you stimulate cells with CXCL6 and one of the most important profibrotic factors, TGF-beta, you can see that there's even more collagen. And so CXCL6 is not just important as a biomarker, but we think it's actually relevant to the pathogenesis of this disease. And so why is that important? We think that CXCL6 works by a particular mechanism, and that's to stabilize collagen mRNA. So mRNA serves as the template for collagen protein, and your cells constantly make the template and break the template, make the template, break the template. And one of the ways to increase the protein is actually to keep the templates around longer. And so this is how CXCL6 works. What you can see here on the left is a time course. Cells were stimulated with a substance called actinomycin D. When cells are stimulated with actinomycin D, transcription stops, or there's no new messenger RNA. And so the template isn't being made. But when we stimulate cells with CXCL6 in the presence of actinomycin D, and that's in the blue curve on the left, is that you can see that there's a tremendous stabilization of collagen mRNA. And so we think that the collagen mRNA in the presence of CXCL6 hangs around longer and is translated into protein much more efficiently. On the right, you can see that TGF-beta does not do this. TGF-beta does stimulate collagen, but by a different mechanism. So we think that CXCL6 is very important. And so how are we going to study that? And so this is funded by the Department of Defense. The Department of Defense has given us an opportunity to try to develop this technology, which might have been seen as risky, but we think is going to identify new therapeutic targets. So I work with Dr. Andrea Berman in the Department of Biology and my close colleague, Dr. Hari Bahu-Dhanapati, and we have devised a way of tiling these templates. So what this does is we take the collagen mRNA, it's in the mixture of cells, we can tile it with different small regions of complementary RNA. And these tiling regions of RNA bind specifically to the collagen mRNA or to the collagen template. And we're able to on these regions of mRNA, use a substance called biotin and then pull down these collagen mRNAs. And by that, we can specifically pull down the collagen mRNA, and we can identify all the proteins that are attached to the collagen mRNA. And then we can send that stuff to the mass spectrometry and can identify all the new proteins. And so I can tell you that we've identified some of the proteins that have been known to bind to collagen, including poly-C binding protein or LARP1. And these have all been studied before, but we've identified many new targets. And you'll have to come talk to me afterwards if you want to hear about the new targets. But again, this is funded by the Department of Defense. And so this is a collaboration with researchers who are not in the pulmonary fibrosis field, but have ideas that are potentially relevant to pulmonary fibrosis research. So in conclusion, you know, the DoD research is directed by acts of Congress, okay? So it's important for all of you to write letters or to call your congressional representatives. One is to keep the government open because I think you'll be furloughed if the government stays open, and so they have critically important work to do. But you tell your congressional representatives that pulmonary fibrosis is important to you and to ensure that the CDMRP continues to fund pulmonary fibrosis research. Two is that the DoD has funded high-impact research in IPF and scleroderma and rheumatoid arthritis. And so again, for the researchers out there, you gotta be in it to win it. And I think it's very important that people consider working with the DoD. The DoD, as Matt reminds me, is not a public health service institute, and so it's possible that projects can be reviewed that are also under review at the NIH. And so you can parallel process to try to get these projects funded. And again, the direction of research is impacted by consumer advocates. So they're highlighting the importance of being involved in the work of advocacy organizations like the Pulmonary Fibrosis Foundation. And so I encourage all of you to lobby your congressional representatives and to remain involved in pulmonary fibrosis advocacy. And I just want to say again that it's a privilege for me to continue to work with this village. Thank you.
So thank you. It's great to be here. And this will be really another story about advocacy and another story about an incredible village. I'd like to say that the Pulmonary Fibrosis Foundation has been a lead in this effort since the onset. So there is an urgent need for reform, and I'll try and walk through in 15 minutes what really has been a journey and try to give some insight so you kind of understand the backstory of the incredible efforts of working with Medicare and legislatures, and the people who have been doing that are really the grassroots patients and professionals. It's been a joint effort, no conflicts to disclose. So I think really the need for reform is come at the time of, I called it the perfect storm. There's really 4 issues that I think contributed to where we are today. One is a major decrease, and I'll go through some of the funding changes in Medicare, or Centers for Medicare and Medicaid Services. Over the past 15 years, there have been dramatic cuts. And in combined with that, there's been minimal change in our oxygen device technology, and we await for some of the NIH funding to. I'm going off again then. And then we had the COVID pandemic right in the middle of this, kind of towards the end. And then we have an ongoing and increasing number of patients with chronic lung disease. So before I delve into this, we need to all be on the same page, a little bit of 30 seconds on Oxygen 101 in terms of devices. So we have 3 choices. There's compressed gas. This comes in tanks. This is any size. It can be portable on a backpack, or it can be the ones in the trolleys that are not so portable and weigh about 18 pounds. Our second option are what we call concentrators. This is a device that takes oxygen out of the ambient air and concentrates it. You can see there we have stationary devices and portable devices. This is the only device that can go on an airplane that's FAA approved. And you can see in the bottom of that circle, one of the stationary concentrators has an apparatus on top of it. You can actually put a compressed gas tank on there and fill it at home. We call these home fill units. And then the third option, which is rarely available today, and we'll talk about that, is liquid oxygen. So liquid oxygen is oxygen that's been cooled down to minus 300 degrees, so it occupies a much smaller space, so you can fit a lot more quantity of oxygen in a smaller container, and it becomes gaseous, of course, when it comes out. And you can see it comes in both stationary and portable modes. So it's important to know this because you can imagine when we walk into legislators' offices and start to talk about oxygen, they're glazed over, they have no idea, and there's a lot of technical information to get across. So some of the historical financial issues that have really resulted in a decrease in reimbursement from Medicare to our durable medical equipment companies, the DMEs, started back in 1997. With the Balanced Budget Act, which decreased oxygen reimbursement by about 25%. And then in 2003 was the largest Medicare overhaul that decreased durable medical equipment payments by $7 billion over the next 10 years. And then we had smaller changes in 2004 and 5, new requirements that were really putting more burden on providers for medical necessity and exercise testing. And then in 2009 was really a key turn when competitive bidding was instituted, which really impacted what DMEs were reimbursed for oxygen services. So competitive bidding defined interested vendors send their bids for consideration within a specified period. The bids are sealed to increase confidentiality of each vendor's proposal. And after the expiration of the bid submission period, the recipient of the bids selects the bidder with the lowest or best terms for the requested fee for that service. So it's a bit like, you know, you're redoing your home in somewhere and you go for a contractor and you're trying to get the lowest bid, but you do want to have a nice looking end product. And really what happened, some of the intended effects was definitely to create competition, pricing among suppliers to help bring costs down. The idea was to set up a single payment based on an accepted bid and its award for 3 years. That's why you'll hear some patients, and many of you in here talk about that first 3-year period. Once you're into that period, you can't switch DME companies. It's very difficult because of that. One intended effect, of course, was to save Medicare program money. There had been reports of abuse and fraud and needed to be reined in. And it was hopefully to ensure, of course, access to quality service and items. So in 2016, Medicare issued a press release that the competitive bidding program continues to maintain access and quality while helping to save Medicare millions. In the first year, CMS estimated that the program saved $200 million, which is a drop in expenditures over 42%, particularly in the 9 different markets that were participating in the program. They expected to save another $25 billion up until last year, 2022, because of this program, and that beneficiaries would also benefit with savings. So one of the things that we saw fall out from the competitive bidding, the first thing really was liquid oxygen decreasing. And I'm not sure you can see it clearly, but you can just look, I think, at the patient column is the most telling data. This is for stationary liquid. In 2010, there were 32,000 patients that were using stationary liquid. But by 2016, that number went down to just about 6,000. And for portable, it was similar. In 2010, there were 40,000 patients that were using portable liquid oxygen, and by 2016, this dropped to 8,000. So Medicare, conversations with Medicare trying to document the problem that we were seeing, lack of access. This is their data, so it was very clear that there was a drop. And it wasn't as if our chronic lung disease patients were improving or decreasing in numbers. There was no change. So the other important points about why liquid oxygen is difficult to access because of the reimbursement issue is it's costly. And the trucks are regulated by the Department of Transportation. They're very specialized. The drivers have to go through special training. If the reimbursement, just to give you as an example, prior to competitive bidding, it may have been that DME received $250 per patient per month for oxygen equipment. And it's important to understand that the Medicare reimbursement does not cover people or personnel. It only covers the equipment and what goes in that, so the tanks, the cylinders, and the gas. So they have a weekly delivery to go out to liquid oxygen patients because they need their reservoirs filled every week, and it's a $40 to pay a driver to go out once a week. They've already almost used up their monthly reimbursement because it had fallen from about $250 a month down to almost $125, and that's just an average, but it was a dramatic decrease. And again, you can see this is data back to 2004. So the liquid oxygen users dramatically went down. So Medicare continued to state that because the DMEs had signed a competitive bidding contract and it included liquid oxygen, that they, of course, should supply that. But in reality, they weren't supplying it. And as providers, we could not get that to our patient. And in some cases, patients were being informed that it was being removed from their home. So DMAs, I mentioned the monthly fee was very challenging. And what we were seeing was a reversion to home fill units. So they were switching patients from liquid to a home fill unit, providing them a few compressed gas tanks, and they never had to go back to that patient's home. So it was cost effective for the DMEs. It's another important point to understand that the portable oxygen concentrators don't deliver more than 3 liters of continuous flow per minute. And there's very few that actually deliver that amount in continuous flow. And those weigh about 18 to 20 pounds. The most common POC, even at a setting of 6, does not deliver more than about 1 to 2 liters per minute. And this is something that we've had a conversation with the FDA about a month ago, talking about the marketing and education that these POCs are being sold to patients or provided to patients. And it's not clear that setting of 4, 5, and 6 does not equate a liters per minute of oxygen delivery. That's a whole nother talk. So in 2015, I chaired the Nursing Assembly and the American Thoracic Society Planning Committee. And we actually were in our room meeting all day. And what it turned into at that point, this is about 5 years into competitive bidding, was us absolutely overwhelmed with patients that were dealing with lots of barriers to getting their oxygen and delivery problems, lack of access to high flow. And so we needed data there was no data. And this was the first really large survey of almost 2,000 patients that we published. It was an electronic survey. It went out to every state was represented in the US and completed, and that was published in the Annals. So this is just one piece of the results, that 50% of patients reported, yes, we do have problems with our oxygen, and this were the types of problems they reported. And what we were very surprised was that over half of them reported that their equipment wasn't working. And then you can see travel with oxygen problems, delivery problems, lack of portables I can manage, lack of high flow portable systems, not enough tanks for activity outside the home. And this is really one of our real efforts to highlight mobility, which is a challenge with carrying around oxygen tanks. And you can see the multiple issues that patients, and many of you in this room, have reported to us. So after that data was published, CMS, we agree that individual problems with access to items and services may not be detected in the claim and health outcomes monitoring, but we don't agree that widespread issues exist that are undetected. So that was a response to their public comments. So what I'm really going to highlight going forward here is the amount of effort and collaboration that came to address educating Medicare, gathering data, and collaborating with them, as well as with legislatures. So you can see kind of the trajectory in 2018. We still needed to provide them more information. The COVID hit, and the PFF was involved in surveying our care center network medical directors and documenting oxygen shortages, and that sometimes we couldn't discharge patients because there wasn't available oxygen. So in 2021, there was reports to CMS, a request to CMS to report on their access and trends. And the CMS did acknowledge that there were still issues that they needed to highlight. And this is another ATS-funded project that at this point we really, and this was a real turning point, this was around 2018. We gathered together stakeholders from all aspects, and we had the FDA there, the NIH, CMS was there, we had every foundation, PFF, American Lung Association, Pulmonary Hypertension, COPD, I know I'm going to forget somebody, and this is published. And then we had nurses, physicians, respiratory therapists, patients. We had academic physicians, community physicians, and we were in an airless, windowless room for 8 hours, and basically had 4 groups of stakeholders to represent everyone's viewpoint. We all had to stand in the footsteps of those, and we had a DME company. We had the head of APRIA there as well. So we had industry, we had patients, clinicians, and the providers. And this was published, and findings of the workshop confirmed the existence of significant barriers to optimal oxygen therapy from all stakeholders' perspectives. So again, it was about educating about devices, about how do patients get out of the house with enough oxygen. We have patients, young patients, pulmonary hypertension and LAM, which is a rare lung disease. Some of these patients are caring for children. And so we had to, a lot of education around, why are we here asking for additional funding so that this patient can get 4 extra E-tanks so that they can take their, either spouse or child to a physician appointment or rehab. And you can see, this is one of our LAM patients on the right. This is her oxygen for the day to come to our clinic. So I've been on the Hill. I think the Hill is like a different country. It's got its own language, acronyms, and it's fascinating. But it's a very, and for this project and advocacy work, it's been a very ongoing and consistent messaging, meeting with legislators, educating them, and the patient and the community. The PFF has had Hill Days. So this has been an onslaught to get the message so that there's an awareness on the Hill. It's kind of the first step. I'm not going to go through each of these letters, but you can just know that there was a demand for reform that came from all aspects. This was in 2018, a letter to Health and Human Services that the health of patients is at risk because of quality and availability of supplemental oxygen. Another letter to CMS that access to liquid was diminishing, DME services were diminishing. And then a letter to one of our representatives from a patient who had liquid oxygen removed from her home. Again, PFF was a leader with many other organizations submitting letters, and this is what the letters looked like. And then we did publish guidelines, which had not been done before, and one of the recommendations from the guideline, really a clinical recommendation, was that patients who had high flow needs, which we defined as over 3 liters continuous flow, and needed to be out of the home, needed liquid oxygen. So we've really regressed. I mean, when I did pulmonary rehabilitation in the 90s, we had liquid oxygen. Patients came with their containers on 10 liters a minute, continuous flow, would fill from our tanks, do their exercise, fill up again, and it was portable, and it was high flow. We are now back to 20 years ago, so we need those devices. And it's really frustrating for the patients, primarily, to lug around this cumbersome, as the patient, again, had to use 2 e-tanks. So where are we today? So because of this really ongoing efforts from patients and advocacy groups on the Hill and the messaging, we now have a formal legislative language proposal. And we've called this the 4 pillars of oxygen reform. And I say we, after that workshop, the ATS granted us a permanent special interest group. So I chair this oxygen special interest group. And we meet the second Monday of every month, and there's 35 of us, and it's exactly that, multidisciplinary, multiple stakeholder representatives, and messaging updates, very formatted approach to this. So this is the result of that. And the Lung Association's been instrumental. You know, some foundations have paid lobbyists full-time on the Hill, some have offices in DC, and some have none of that. It is volunteerism. So number 1, ensure that oxygen is patient-centric. Change home oxygen to supplemental. It actually takes an act of Congress to change the CMS benefit title from home to supplemental. Create a patient's Bill of Rights, ensure access to liquid, create a statutory service element that will cover the personnel and the professional respiratory therapists, and establish a national document, a standardized document for an order template. So as of September, we have a Senator Cassidy who has agreed to take on this legislation from the Republican side, and we are in the process of finding a Democratic representative, so there's bipartisan support. And that 4 pillars document is now being drafted for a legislative proposal, and hopefully we'll have a bill number that, once that is identified, we'll be asking for everyone's support. So I just want to emphasize again how many people, and I say I have American Thoracic Society just because that's kind of been the home for this, but the PFF has been a leader from day one. They have created outstanding oxygen educational materials for patients. They have a support line, 1-800-OXYGEN, that patients can call. But it has taken a village to get, this has been a long haul from 2015 when this started for me. And these are all the organizations, you know, adults, pediatrics, inpatient, outpatient, that oxygen really impacts. So we are continuing. This is just an onward march for many people. These are our patients who are definitely outside of the home. And I would end with this as one of our PHA, tireless advocate on the Hill. And she was actually at an American Board of Internal Medicine pulmonary meeting, and this is her portable oxygen and the way she had to travel for the day. So we really thank our patients for the amount of work that they have helped drive this legislation. They've written letters, given examples, and it's really the only reason we're here where we are now. So thank you very much.